you find helpful? Yeah. Okay, we'll keep doing it. Okay, so does everybody understand what mitosis is? What is mitosis? Before we go on, I want you to explain it to me. Correct. So it's a cell making what? Two daughter cells. That's what? An exact copy of itself. So what has to be done in order to do that? Yeah, we have to make a copy of the map, genetic material, because that's going to direct my cell's function. And then what else? Copy of all the organelles. We have to do that too. And then we have to split it apart. So the process of mitosis involves organizing all that copied genetic material and getting it all lined up nice so we can split it apart. And we want to make sure what? When we split it apart. Correct. So we want to make sure the copy that we give to each of our new, what do we call those new cells? Daughter cells have the exact same genetic information so they can carry on the exact same function. That's mitosis. Okay, I thought that was mine. I was going to flip out. Kill it. Sister chromatid, we're going to start talking about here. Sister chromatid are also copies of what? Yeah, the chromosome copies. So when I make two copies of the same thing, they're sisters.
Okay.
Does that make sense?
Okay. So then the daughters are going to be...
The daughter cells are the two cells that we create. Does that make sense? Yeah, sister and daughter. We probably should have. We probably should have used different terms, right?
Because they were using two different ones. Like at one point, I think we were talking sisters, and all of a sudden we'll start creating daughters.
Right. It's like, oh shoot, is that the same thing? So that's a good question. No, the sister chromatid is what we refer to as the copy, and then the daughter cells are the brand new cells that are born from the first one. Gotcha. Okay, when we talk about reproduction, the same thing has to happen. In order for me to make reproductive cells, I have to make a whole bunch of them so I can make a whole brand new individual. But when we talk about sexual reproduction, we have to remember that it involves two separate parents. And here's where we're getting confused with the whole parents thing. Two separate organisms, let's call them, that are going to contribute to the brand new organism that is produced. So, life cycle of a multicellular organism is the different stages that are going to lead up to adulthood and the generation to the adults of the next. So in order for us to continue on as a species, what do we have to do? Because some of us think we're the end all and be all, and that's it. I'm here, we're good. That's not the case, right? What do we're only going to last for a very short period of time. Yeah, we need to reproduce as a species. So when we're talking about sexually reproducing organisms, we're talking about the union of cells from typically a male and a female of the species to come together to create a brand new organism. In order to create the cells that are going to combine to make you, how many chromosomes do you have? 46. I have to create reproductive cells that only have what? 23. Remember when I drew that stuff on the board last class, we saw that we have two copies of information for every single trait we possess. And we got those from our parents. So we have to make what we call a haploid cell, a cell that has half of the genetic material as a somatic cell. Somatic cells have what? 46 chromosomes. These haploid cells, or another name for them are gametes, have how many chromosomes? 23. We also see the letter N. So if I have 46 chromosomes, I also have what with respect to N? 2 pairs, or 2N. If I'm a gamete, bless you, a reproductive cell, I have 23 chromosomes, or N. So we see haploid cells, N. If they are female, and we're talking human here, they are eggs or oocytes. And males are what? Males' gametes are called what? Sperm. When they come together, fertilization takes place, and that one brand new 46 chromosome cell is the beginning of a brand new organism. You with me? Good. Oh, wrong one. So humans are what we call diploid organisms, because their cells, most of their cells that are possessed in their body, have 2 pairs of information for everything that they need to do as a cell. So there's 2 sets of chromosomes for every single trait that you possess. Haploid cells are gametes, have half the information. So sperm and egg come together during fertilization to form a diploid cell, and that beginning cell, or the beginning few cells, are called zygotes. So when your fertilization took place, you were a zygote first. And after about 3 months-ish, well, then you turned into an embryo, as you got a little bigger. And then what happened to you after about 3 months? You're a fetus then. So there's different names depending on what stage you are in development. So sexual life cycles involve alternation of diploid and haploid stages. Meiosis is the process, the cellular division process, that's going to create those haploid gametes. Basically, the difference between meiosis and mitosis is meiosis is a 2-step process. During the regular mitosis, when did I replicate my genetic material? Interphase, right? During the S phase of interphase. That's when I made the copies of my genetic material. Basically what's going to happen is that exact same thing that we saw in mitosis, when reproductive cells are going to start to be made. They're going to go through the whole mitosis thing, and then they're going to kick in and do it again. But what are they going to skip? Yeah, they're going to skip that interphase part and go right into meiosis, or mitosis again. So that they're going to split the same exact way, same exact stages. We're going to call it meiosis 1 and meiosis 2. But we're going to skip the copy part. So the end result from one cell is going to be, so we have one cell in the beginning with 46 chromosomes. Then it was going to do what? Make what? Make 92, and then it's going to split into this, yes? Then that's going to split into, because we're not going to do this. Yes? So, That is the making of a gamete. So in your reproductive organs, females, what are your reproductive organs? Yeah, making of cells, the reproductive cells. I'm trying to make this simple. What do you ladies have? Where do you make your reproductive cells? In your ovaries. Males, where do you make your reproductive cells? In your testes. There are cells that sit and wait, ready to become these guys. And when they start the process of making these guys, this is what they're going to do. Except we call that two-step process meiosis. Okay? And we're going to call it, this has the same exact stages. So there's a prophase, and there's a metaphase, and there's an anaphase, and there's a telophase in meiosis as well. So interphase before meiosis, the making of gametes, is very similar to what we already learned. The same exact thing. Interphase. This guy's sitting there waiting to become a reproductive cell. So he's doing what he's doing for a living, and then all of a sudden he gets the chemical signal. And that's going to cause him to do what he would have done if he was just going to split. Replicate the genetic material. In here, we see a pair of homologous chromosomes in a diploid parent cell. What's a homologous chromosome? Have the same what? Not the same DNA necessarily, but the same codes for stuff. Remember the picture I drew up here? One's for hair, one's for hair, one's for eyes, one's for eyes. You all have two, right? So we're going to duplicate. Go from here to here. Make my 92. So we're going to duplicate the pairs of those homologous chromosomes. During meiosis 1 and meiosis 2, we're then going to split the pairs. We're then going to split into 46, meiosis 1, and before we get a chance to replicate, we're going to split in meiosis 2. So that the end result is going to have half the genetic material as the parent cell. Okay? Sometimes that picture can be a little confusing because you see two up there. But remember, you start with two pairs of everything. Yes? Go ahead.
I guess my confusion comes from, because if you go into one of these pages, they separate the colors into boy, girl.
Yeah.
So here, right now, we're looking at just one person.
Two copies of information.
Okay.
Okay?
So that's,
Yeah. It's not boy, girl. Just think copies of everything,
yeah,
like
I
eye
understand
colors
that.
and...
You know, once you get reading further in here, then they break it down into, pretend that this is the blue is the male and the red is the female. And
Yeah.
this is the
Yeah, we're not, so yeah, this, yeah. We should have used different colors in the book, huh? Would have been a little less confusing. Are you with me, though, on this? Okay. So, in words, what we just said, haploid daughter cells are produced in diploid organisms. Organisms that have two pairs of information for all of their traits. And each of those traits, let's call them something. There's a word for them. The stuff I have for eye color and the stuff I have for hair color. All of those little pieces of information are called alleles. And I think we used that term last class. Yes? And you have two of them for everything. So meiosis 1, meiosis 2 looks exactly the same, except by the end of meiosis 2, because I didn't replicate the genetic material after meiosis 1, I'm going to end up with haploid cells. The other thing that happens during the making of these haploid cells, and not to be confusing, but it might be. Well, let's try to use some colors. Oh, all I have is green. That's not helpful. We're going to mix up stuff. So remember, you got one copy or one allele from your mother and one allele from your father for whatever. And on each chromosome, there are many different alleles that you received. When you first received them, you got all of one from mother, all of one from father. And then what happened was the genetic material came together and kind of shuffled around a little bit as cells were reproducing themselves. So if I had a different color marker, let's put them in the other direction. This one would be from mother, and this one would be from father. What happens is some of these will go like this. This one will come over here, and that one will come over here. And then maybe this one will go over here, and that one will come over here. And maybe this one will go over there, and that one will come over here. Each strand still only has one copy of whatever it is I'm talking about, but it doesn't stay segregated. It doesn't stay mother chromosome, father chromosome information. It gets mixed up. And that's what they're talking about when they talk about crossing over. So here we see during meiosis 1 all of the things that we saw before: prophase, metaphase, anaphase. We see our chromosomes replicating. We see them splitting. But now they look a little bit different. You see what happened here? Now if you look at the picture, the pairs are still the same. They're going to line up, same traits. But this one's red and blue. And this one's blue and red. What happened? Yeah, crossing over. They still possess one of each trait, but it's not evenly distributed anymore. This is why when we take two organisms and we bring them together, their offspring are so diverse. The more we can shuffle up that genetic pool, the better. It's why you're not supposed to marry your sister. Yes? How come? It's a little creepy, yes. But what else? Why else? Why is that a law? Why is that something we as humans have established in our laws?
Because our inbreeding.
When there, no, not trial and error. When there are mistakes in an organism and you breed them very closely, and you might have seen this if anybody ever bred dogs or any other animals. When there are mistakes, and there's always lots of mistakes that can occur in our genetic information, when you breed too close, you're going to enhance those mistakes. Mistakes like problems with nervous system, mistakes like problems with, think about dog problems. Anybody like dogs? Dog breeders? Why don't you want to breed them very close? Because when they're bred very close, mistakes are enhanced. Things like hip dysplasia in Newfoundlands, for example. When you breed the dogs too close, those mistakes are enhanced. So it's very hard or very important that we mix up our gene pool, basically, so we can be hardy as a species. So meiosis 2 then, we can see all of those little chromosomes all mixed up now, are going to go through the exact same thing right after telophase 1, the first meiosis split. Again, not giving the chromosomes a chance to replicate, they're going to enter prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. Same exact stuff. Except the end result is not going to be something that looks exactly like the parent. It's going to have what? It's going to have half the genetic information, and what else? That crossing over has taken place. So we're going to mix up the gene pool a little bit. Is that a good thing? Yes.
I just have a clarification for you real quick.
Yes, sir.
This has always like confused me about like meiosis. How is it possible to have only 23? Because like how do you know you have like all the alleles you need? Like, you know what I'm saying? Like, what is if like
And what? Yeah, go ahead. Keep going.
I don't know how to explain it. Like,
What if you don't have all the alleles, or what if the split didn't happen right? That happens all the time. What's that called? Yeah, it's called different diseases in humans.
like,
What if I get
the
too
whole, like,
much or not enough?
like thing, but like, how do you know that like?
Once the organism, if it's,
How to, like, word it
if,
out.
no, I know exactly what you're asking. Once the organism develops, if it's able to develop, you're going to see the results when they are born and they grow up.
Right.
So you're going to see deficiencies, you're going to see diseases, you're going to see abnormalities when the genetic information wasn't passed on properly. And that happens all the time.
Oh, I know, I got that. Like, I just, what, I don't know how to word my question. Like, I feel like you know what I'm saying, but like.
Yeah, I do.
But you don't at the same time.
But sometimes there's little things that you'll never know.
Yeah.
You might be missing or have too much of. But there's certain ones that will manifest itself as diseases or abnormalities in the adult.
There's a lot of tests now that test for that, mutations or defects.
Well, aren't you supposed to get your blood tested before you have a baby?
No, not anymore.
There was,
Yes.
did you have to get, a while back, did you have to get your blood tested before you have a
If your mother, if you are, and that's a whole nother discussion. If you are an Rh negative mother, yes. It's a protein on the surface of your red blood cells. And if you mate with an Rh positive male, you have to be very careful that your child is an Rh positive and you don't make antibodies against that antigen. And we'll talk about, we're going to talk about that. So.
Is RH negative. She has to have, she had shots for each child.
Yep. And the shot is basically antibodies against the antigen. Because when you give birth to that child, no matter how good the process goes, child's blood is always going to mix with your blood, introducing the antigens, foreign substances, into your body. If you don't get the shot, which is basically a whole bunch of antibodies against that antigen, and that's called ROGAM, you're going to elicit an immune response against that antigen. So the next time you get pregnant, if it's another Rh positive child, your antibodies will attack its antigen during the pregnancy because those antibodies are small enough to pass the placenta. If you never make them because you got the ROGAM shot, you're all set.
Yeah, you have to get it while you're pregnant and after the baby's born if they have Rh positive blood.
Correct. And they do it anyway, no matter what. They don't even take any chances. Because they don't want you to ever make those antibodies. You don't make them naturally. So does that make sense? Probably not right now. It will when we talk more antigen antibody.
All right, so you have two alleles for each trait, like let's say eye color. You have two alleles for each trait?
Each trait except if you're a male.
Right.
And they exist on the X and Y chromosome.
So each, like you have two pairs, so in like each gamete, they're split up. So like you have 46, right?
Correct.
And you have two pairs of hair color alleles in 46. So they would be split up.
Correct. So one hair color goes to that guy, and one hair color goes to that guy. But that guy's going to mate with this guy. And he only has one.
Yeah.
You get it?
Now you got two again.
Then when you get two again, you get a whole brand new person. Yes? Can that cell become a whole brand new person?
No.
No. It's missing half the information. Does that help?
Oh, I got it.
Yep. All right, so mitosis and meiosis, the chromosomes duplicate only once during the preceding interphase when we talk about meiosis. So interphase only happens at the very beginning, and then we have 2 rounds of splitting in meiosis. Mitosis uses 1 division and produces 2 diploid cells. Meiosis uses 2 divisions and produces 4 what? Haploid cells. Make sense? All the events unique to meiosis occur during meiosis 1. So, we see the differences between meiosis and mitosis. Mitosis, parent, split, kids look exactly the same. Meiosis, 2 rounds of splitting, and what happens to the chromosomes during the process? They get jumbled. The crossing over happens. So we get all kinds of weird combinations down here in the gametes. Yes, dear?
So meiosis 2 only happens in female
And male.
when they're producing an egg.
Correct.
It happens in the male when the male is ejaculating.
Or producing, no. It hopefully happens before that when he's making sperm cells. Yes.
So right before you ejaculate.
It takes a little longer than right before. You're doing that all the time, fellows.
So what happens?
And guess, then they die. And they get reabsorbed. What happens when you have a vasectomy?
What happens when?
What happens when you have a vasectomy? What's a vasectomy?
I don't know.
Sorry, yeah, guys don't want to talk about anything. You still make them constantly. You slow down in your production as you become
So
less used.
Anyway, even though they only drop an egg every so often.
Yeah, it takes 28 days to create one.
I think that was almost what you guys said.
I don't understand. It's so funny. I'm trying to.
No, but you're right. Do you understand? Do you understand? Okay, so you're doing that all the time. Males, when ejaculation takes place, do you know how many sperm there are in the ejaculate?
Brazilian? I don't know.
2 million.
Well, my
But this process takes a few days.
cell is going to go through meiosis 2, right?
Only when you're making sperm, yes.
But a female isn't going to go through meiosis 2 unless she's dropping an egg.
But it takes her longer to make the egg.
28 days.
Correct. Takes you 2 or 3 to make 2 million.
That's why we're the man.
No, we get it right the first time. Okay, so the offspring of sexual reproduction are genetically different. They have the same pair of everything, but they're going to be very different from the parents because of what? That crossing over thing. So every time a female makes a gamete, there's tons of crossing over. So even though it's got half, it doesn't have exactly half. It has kind of half. And the same thing happens with the male and the sperm. So our offspring can be very, very different than the parents. Not only because you have 2 different parents, but now you've mixed up their genetic material even more. Yes?
How do you tell if certain parents if the codes can be rearranged?
Because there's certain genetic information that you get directly from your parents. For example, the DNA you get in your mitochondria comes from your mother. And only your mother. Certain patterns in genetic information will only come from your father. Okay?
You said the patterns can be crossed over.
Correct. But there's certain just think about throwing a dice. There's 6 possibilities you can throw, right? And then those 6 are common to the dice. Yes? Even though they might be in different arrangements, and they might actually have different results, you're still going to have the 6. Does that make sense? So, when we talk about getting our chromosomes from our parents, we talk about something called independent assortment. And that's because of the whole crossing over thing. They're going to independently assort. They're not going to be one for one. My mother's egg is not going to be only from her mother. It's going to be a combination of her mother and her father. Yes? So, every chromosome pair orients independently of all the others at metaphase 1. For any species, the total number of chromosome combinations can appear in gametes due to independent assortment. So 2 to the nth power, where n is the haploid number, are the different mathematical possibilities. So you ready to blow your mind? For humans, n is 23, right? So with n being 23, how many different chromosome combinations are possible just in your gametes? That's not including bringing together 2 gametes. How many different possibilities? 8,388,608. It would be a hell of a hard time to win that lottery. Yes?
Is this our bonus question of the test?
No. It just gives you an appreciation for why we're all so different. Yes?
So that's 8 million, but there's like 6 or 7 billion people. So is there somebody out there that's like...
Yes.
Possibly. Yep.
There's like 6 people in the world that have very similar features to you.
Yep.
Oh, I want to
You might have almost the exact same genetics as you.
It's not going to work. You're not going to get any more powerful.
But think about it. Think about this, okay? If it was that easy to guess the exact combination in the lottery, why doesn't everybody in the United States win the lottery all the time? And how many numbers are we talking about there?
6.
No, how many? 36 numbers, right?
There is someone who wins the lottery every week.
So
Just
is it,
for us.
don't you
No,
take
the
the
same
function of
person.
36?
Same person every week.
36 times 35
Week.
is 34, 33, 31. That's how
So
much-
now instead of, we're talking, and we're not talking 23, we're talking 23 chromosomes. How many different alleles are on those 23 chromosomes? The numbers are crazy, yes? So we never know what's the outcome is going to
In
be.
sexually reproducing organisms,
I wish I
3
could get this
processes lead to most genetic variation. Independent orientation of chromosomes in meiosis, crossing over of chromosomes in meiosis, and random fertilization.
Yeah, won't.
Each pair of homologous chromosomes consists of 1 chromosome inherited from the father and 1 from the mother. Here we have color-coded them blue and red. Each pair of chromosomes lines up independently of the other pairs in metaphase 1 of meiosis. Here you see one of the possible arrangements and outcomes. There are 2 different ways that each chromosome pair can line up. That means that in the organism shown here, with the diploid number of 4, independent orientation of chromosomes at metaphase 1 can produce gametes with 4 different combinations of maternal and paternal chromosomes. In a human being, with 46 chromosomes, more than 8 million combinations are possible. Now let's look at how crossing over creates even more genetic variability. During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of 2 non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from 2 parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes. Independent orientation and crossing over occur simultaneously during meiosis, multiplying the number of genetic variations among gametes. Because each pair of chromosomes lines up independently, and crossovers can occur almost anywhere along each pair of chromosomes, it is possible for a human being to produce an almost infinite variety of gametes. A sperm fertilizes an egg, producing a zygote. The random nature of fertilization adds to the variation arising from meiosis. Each parent is capable of producing a huge variety of genetically different gametes. The number of possible combinations among their offspring is staggering. Theoretically, one human couple is capable of conceiving a number of genetically different offspring that is far greater than the number of humans who have ever lived.
Scary, right?
That's pretty crazy.
It is crazy. Yes.
How does this work with like mules? Where it's like, no, because like they're like from a horse and a donkey.
Are they? Can they reproduce? Can they? No, they're not fertile.
But
They can, and this is what happens like interspecies. That's two different species that come together to create a new, but that new one can't make any more. I have no idea. But it's not necessarily going to be a successful. I mean, they can make a new organism, but it's not necessarily going to be a successful outcome. If it was, then that's probably how a new species is going to be able to evolve as well. I don't know these little things. Probably the same thing during, yeah, isn't that was actually made, yeah. I don't know. Somebody's got to Google that. Yes.
Does crossing over happen every single time mitosis happens?
Every single time meiosis happens?
Mitosis, because I mean.
Not mitosis.
But why? Because prophase is the same, isn't it? The first prophase is the same.
Yeah, but for some reason during meiosis that happens.
It must have something to do with sex.
And does it happen during mitosis? Probably.
See what I'm saying? Because like.
Yeah,
I mean,
but what's the
it's
difference?
basically the same thing except for.
What would be the difference if I have these two, and then I give them these two? If it's on this chromosome or that chromosome?
I'm just saying is that yeah, it's good.
We're good?
Yeah.
Okay.
I was confused. I was just asking a question.
But that's a good question. You'd still get the same information. So combination A, combination B, combination C, combination D, we see how many different possibilities there are in our gametes. So staggering numbers. If each gamete represents 8 million blah, 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 different chromosome combinations of fertilizations, humans would have 8 million blah, blah, blah times 8 million blah, blah, blah when two of them came together, and more than 70 trillion different possible chromosome combinations for humans alone. If I have more chromosomes and I'm a sexually reproducing organism, that number's even higher. Yes? So we see the random nature of fertilization, so we can't really ever guess what the outcome is going to be. Not only that, during ejaculation, 2 million sperm. Yes? Tons of different combinations. Who's the one that's going to fertilize that oocyte? The one that's the fastest and makes it there first. So there's even more variation. You know, what causes a sperm to be faster or slower? So crossing over is very important. It's important in mixing up our gene pool as a species and results in genetic recombination. Production of gene combinations different from those carried by the parental chromosomes. So even though they are our parents...
During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of two non-sister chromatids may trade places. This process of crossing over...
I don't know what this one will do for us. think this is a bunch of... So, no scientists have ever found male rotifers, the little single-celled organism, microscopic freshwater invertebrate. Does this entire class of animals reproduce solely by asexual means? Yeah, probably. Research compared sequences of particular genes and found the results to be some of them sexually reproducing rotifers had a nearly identical homologous gene differing by only 0.5%. Two versions of the same gene in asexually reproducing rotifers differ by 3.5 to 54%. So, these little guys have evolved for millions of years without any sexual reproduction. So they reproduce what? Asexually. So what happens when there's mistakes? Mistakes happens all the time. So just think about all shuffling of all this information. Some of them can be fatal. Some of them will allow you to survive, but not so-called normally. Yes?
I was just going to ask, this was talking about last class, but when a sponge buds, is that the same, is that asexual?
I think so, yeah.
Maybe it's like a weird theory.
I don't know, though. I'm not familiar with the sponge's sexual, if it's sexual or asexual.
You need to watch more Patrick.
I know. More SpongeBob. Yeah, and a lot of sea creatures will actually spread their sperm over egg in a water environment, separate from...
There is male and female. Talk about little polyps when the male shoots out their
Sperm.
They meet into the water.
Yeah. Yeah, and fish in general tend to... Females will lay the eggs somewhere and males will come and fertilize them. So it's still sexual reproduction, but in a different way than we're familiar with. All right, so mistakes. Non-disjunction. Members of a chromosome pair that fail to separate properly at anaphase. They produce gametes with the incorrect number of chromosomes. So that's a non-disjunction. We might have too much, or we might not have enough because of their splitting during meiosis. It can occur during meiosis 1 or meiosis 2. So it's never perfect. Sometimes when we get a split, we'll have too much, +1, or not enough, -1. If that happens and I make 4 gametes, say I'm a male, from one cell, and I make 4 gametes, 2 of them are going to be abnormal. Do you think that during your production of 2 million gametes, there's some abnormal ones in there? Absolutely. Same thing can happen in a female. So, depending on what extra material you get or lack of material that you get during fertilization, that's going to determine whether or not the embryo and the fetus can actually survive. And a lot of times when females have miscarriages, a lot of times it's because there's some sort of mistake that wouldn't have allowed that embryo or zygote to fully function. And that happens a lot. It's tough to handle, but sometimes if we think of it that way, it's a little bit easier to handle. And the other thing is it's hard to genetically test every single loss of pregnancy, because many times the child is too small or the cells are too small to retrieve to genetically test. But if we could do that, we would see that quite a bit. So non-disjunction occurs and a normal sperm fertilizes an oocyte or an egg with an extra chromosome. Now we have 2n plus what? We got extra stuff. You think extra stuff would be good, but it's not. If the organism survives, again, if it lives through whatever this non-disjunction is, we'll see an abnormal karyotype. Some disorder or syndrome might be caused by the abnormal number of genes. And a very common one that many of us have probably heard about is a non-disjunction in our 21st pair of chromosomes. A trisomy, we call. So how many pairs of that 21st chromosome do we have? Instead of 2, we have 3. And that's Down syndrome. We see a cute picture of an adorable little baby there. But Down syndrome is a trisomy. So 3 instead of 2. A condition in which an individual has an extra 21st pair of chromosome affects about 1 out of every 700 children born. And yeah, and we're actually seeing, and why is it why do you think it's more than 1 in every 700? I don't think it's as much as 200, but it's definitely more than that.
I was going to ask earlier too, because I read an article that they were trying to link Down syndrome to all the C-sections.
Not necessarily C-sections, but what we see is an increased incidence of trisomies and non-disjunctions like this in older mothers. And what's happening in our society these days? People are waiting a lot longer. Back when it was your grandparents and their grandparents, they started having children at what? 13, 14 years old. Now, waiting till our 40s. Waiting later, we see a higher incidence. So that might contribute to the higher incidence of trisomy. So remember that karyotype we saw in the beginning? We see we have extra material at the 21st pair. Is this individual a girl or a boy?
Girl.
How do you know?
Because there's 2 X's.
There you go. It's 2 X's. Look at the karyotype. Sure it does. Look.
There's 2.
They're both the same size. Remember the Y chromosome was dinky. Well, this is the sex chromosome pair. So they actually should number that what? 23. That's a 23rd pair. So, Down syndrome in offspring of normal parents seems to statistically increase markedly with the age of mother. So that's one of the chances you take. And look at the statistics. Yeah. It increases quite a bit. Most people don't tend to have babies at 50, but yeah, in between 40 and 50 years of age, it increases quite a bit. So. Or realize that you increase your chances of having possibly genetic disorders. Can lead to abnormal numbers of sex chromosomes. So too many or not enough is a problem as well. Perhaps the Y chromosome is very small and carries relatively few genes, but too many of them are going to cause a problem as well. So these are different abnormalities in sex chromosomes. We can see that on page 140 in our textbook. There's a syndrome called Klinefelter syndrome where you have a trisomy of that 23rd pair, 2 X chromosomes and a Y, which kind of gives you a male, XYY, male, and sometimes you don't even know you have that, XXX, female, and then when I'm missing an X chromosome, gives me still a female, but I don't have the ability to have functioning sexual reproductive organs. So I have problems making gametes. So frequencies, 1 in every 2,000 Klinefelters, and XYY, XXX, 1 in every 1,000, and 1 in every 5,000 for X.
What exactly is it?
Studies have shown that males that are XYY, and again, most of the time we don't even know, they tend to be males that have much more problem with rage. And you can go look that up.
I think they call them type A personalities.
No. Like I'm an axe murderer personalities. And a lot of inmates that have
Or
been, or Republicans, that have been genetically tested, who are in there for some heinous crimes, we see a higher percentage of them XYY. So, who knows? I don't know. But you should look, Hannibal Lecter, yeah, the fava beans and your liver guy. All right, so asexual reproduction conveys an evolutionary advantage when plants are sparsely distributed, unlikely to be able to exchange pollen, but asexual reproduction is going to help to do what? Sexual reproduction is going to help to do, well, not asexual reproduction, conserve energy. Sexual reproduction is going to do what for you, though? With respect to genetics, give you more variety. So if there's mistakes, we can kind of, not weed them out, but maybe even mask them for many years, because we inherit a normal set of alleles from another parent. So it might be an advantage because it's speeding up adaptation to change in the environment and allowing a population to more easily rid itself of any harmful genes. So there's pluses and minuses to both asexual and sexual reproduction, but sexual reproduction allows for a much more diverse population. All right? So we good? I'm guessing that's a yes. Any questions about meiosis and chromosomes and puppies? They're so cute. No, they're not stupid. They're labs. I love them. I do too. But people, I understand what you mean. Yep.
It's more like puppies, they're like dogs, more like cats.
Has that one personality.
No, cats are manipulative. Wait on me. Dogs are like, I'll do whatever you want, just touch me. Yo, stop! You're horrible. How could you eat one of those? I couldn't do that. Tastes like chicken. Oh, God, you people are horrible. I'm not going to bring my dog to school. Turn into a hot dog. All right, people have selected and mated dogs with preferred traits for over 15,000 years. Who are the ancestors of our wonderful little black labs and all of those wolves? Over thousands of years, genetic tinkering has led to the incredible variety of body types and behaviors that we know in our dogs today. Selecting for traits, picking out a puppy from a litter that has all the traits that we desire, the perfect ears, the cute coat color. Chances are if we take that dog and mate it with another one of the same species, hopefully, not always, some of those traits that we love will show up in the offspring. That's what they're talking about here. So that's the principles, underlying principles of genetics that help us to understand that. So we know that we have 46 chromosomes, and we know that certain patterns or different alleles of information are passed on from our parents. That is what we call heredity. Transmission of traits from one generation to the next. And genetics is the study of that information, heredity. It started way, way back in the 1800s. In the 1860s, a fellow by the name of Gregor Mendel. Do you know who Gregor Mendel was? He was a monk. And he liked the garden. Loved peas. Out in the garden. Had a lot of time on his hands. He was a monk. What do you want? So he started to wonder about his plants. Why they looked the way they did. And how it is they came to be. Did you have a question? No. Nope. So he started looking, and Gregor Mendel also happened to be a scientist and a mathematician. Wicked smart guy. So he started to look at patterns in the plants that he was growing in his garden. And he, believe it or not, way back in the 1860s, came up with the fundamentals of genetics that we still hold true today. Even though he didn't have any fancy equipment, he didn't even have a microscope. And the principles that he talked about way back when are pretty much, we know to be fairly true today. Yeah. So, garden peas, because they were what? Easy to grow. They grew pretty fast. Different varieties, and they are delicious. And you can manipulate them. Why gross?
I don't like them.
You don't like peas? I never did until I ate fresh peas.
I'll eat them out of the garden.
Yes.
Not cooked, but the moment you cook it, you ruin it.
Yeah, me too. Yeah. Peas and asparagus. It's the only thing in the world that I won't eat. Yep.
Asparagus raw?
I'll eat it raw.
Really?
Yep. I won't eat it cooked. It's something, it has to do with your taste buds and all of that stuff.
Yeah.
Is it true that like your taste and personality change every so many years? Not personality.
Well, your likes and dislikes, yeah. Yeah, because I would never eat peas, period. And now that I'm older, I'm a little bit more open to trying things. And I tried fresh peas, and I like them. Yeah.
I can't handle cooked peas.
I don't like steamed vegetables in general. I like them raw.
Yeah, raws. Yeah. So and that changes as we get older. Our tastes change. Babies, when they're little, sometimes they'll migrate to one certain food, but you always have to keep mixing it up. Don't just assume your child doesn't like something. Just keep offering it, and eventually they'll try it and like it.
My kids would eat it, I just gave them vegetables to chew on. Have carrot, have celery, have broccoli. They loved it.
They didn't get chunks off of them and choke on them, did they?
No,
Oops, a chunk came off.
there was chunks of vegetable all over my house.
Yeah, that is,
But then you ought to see my kid now, both of them.
They like vegetables.
Whatever vegetable you put in front of them, they're going to eat.
Yeah, my guys do too. Poultry. Okay, let's get back on topic now. When I look at a plant, did you know I see both male and female in the flower? So when I look at a flower, a flowering plant, the female portion is what we call the carpel. It's in the center. And the male portion is in the stamen. The pollen is what? It's sperm. And the egg or oocyte of a plant or flower is down in the carpel. So when the pollen covers my car, or that doesn't help with fertilization of plants, unfortunately, but the pollen very close to the top of the carpel will come off. And who's going to help it come off and get to the top of the carpel? Different insects are very, very important in the pollination process, right? Bees, when they go in for nectar, well, that's why flowers produce it. It doesn't do anything for them, but it attracts insects. And when insects come to eat the nectar, they brush up against the stamen and transfer that material to the carpel. So fertilization will take place more frequently when there's insects to help with the process. Or if Gregor Mendel decides he wants to control the whole process and cuts off the little stamen and manually goes and fertilizes his flowers. So that's how he did it. That's how he controlled this experiment.
So it's possible for a plant to fertilize itself?
It most always does.
Really?
But not necessarily, because if that bee comes along and gets a whole bunch of pollen all over its little fat legs, that flower might self-pollinate, but then he's going to go to the next flower with the pollen all over his legs and get some in that carpel. And there we have what? Cross-pollination, genetic variation. That's why flowering plants have so many different kinds, because of insects that help them do that.
Is every plant, do every plant have like male and female parts?
They're all a little bit different, but for the most part, yes.
So is that technically asexual?
No. Sexual reproduction is the union of egg and sperm. It is not the act. Yes? Good. So, this is how Gregor Mendel controlled his experiments. See, he could actually go and snip off some of the different stamen and transfer pollen to a carpel of a different plant. That would help pollinate, and then the fruits that came off and the flowers that came off and the characteristics that resulted when he cross-pollinated are the things that he used for the basis of the rules of genetics that we still use today. Now, he didn't know what the heck he was doing as far as DNA went, but he knew that there was some sort of particulate matter that was being transferred from one organism to another, and he called that characters. So a character is some sort of heritable, I can't even say that word, some feature that varies among individuals. Again, he didn't know what that was. He didn't know it was DNA, but he knew we used to call them traits or characters. A trait is a variant of a character. So if I have a plant that has a white flower versus a plant that has a purple flower versus a plant that has a yellow flower, those are different traits. Each of the characters that Mendel studied occurred in two distinct traits. So Mendel created pure varieties that always looked the same. He controlled the environment in which those plants lived. And then he would take purebreds that he had created for many, many seasons and cross them and see what happened, to see what the different traits were in the offspring. When I take one plant and I cross it with a different plant, I create something called a hybrid. Anybody garden? You buy seeds? Would you ever read the package?
Yes. Hybrid.
Have you ever heard that word before? Yeah.
Yes.
Monsanto's took care of all that.
Oh yeah, that's a whole nother can of worms.
Wait, what happened?
Yeah, no, we don't want to go there.
It roses the
Are you
same.
serious?
Yeah. So you see, when you read the package, you see hybrid. Okay.
That's messed up.
Not in the world, just
Just where it's important.
yeah. So watch a movie called Food Inc. and you'll understand. It's online.
How long is it? Like 3 hours?
No.
You'll never
It's,
eat again.
yeah, but it explains the whole Monsanto
Am I gonna
situation.
want to eat this? Am I gonna want to watch this?
You might want to change the way you eat.
Yeah, you will.
I don't know.
All right, so the parent plants, the pure plants that Mendel created, I self-pollinate myself for many, many seasons, we call the parent generation, or P generation. The hybrid is going to be the offspring from these parents. They're called the first familial generation, or F1. And then if I take the F1 generations and I cross them, we have the second familial generation, or what? F2. So the reason they introduce this is because you'll see it a lot as we go through the crosses. So he performed a whole bunch of experiments and tracked different inheritance and characters and tried to stick to two alternative forms. So if he was looking at flowers, he would take two different colors, white and purple, or white and green, or whatever. If you look on page 147, you'll see some of the things that he looked at in his garden. And he found that certain traits tend to, what he called, dominate over others. We have them listed here for the traits that he looked at. Dominant color in plants, the pea plants that he studied, was purple. Recessive was white. The way the flowers grew on the plant itself, axial versus terminal, on the side versus on the top. The color of the seeds in the fruit that was born from these peas, either yellow or green. The shape of the seeds, round or wrinkled. Yep, they exist. I don't think they're very common, but you find yellow pods sometimes. You'll see them. Beans is typically what they sell during the season. The pods themselves, whether they're inflated or fat or constricted around the peas. The color of them, green and yellow, and whether you had tall plants or small plants. Dominant, recessive. What the heck does dominant and recessive mean?
Genes, like, my parents are both blonde and red, and I'm not. So, the blonde is dominant in my parents, and the brown is recessive, because out of my family, I'm the only one with dark hair, everyone else is either red or blonde, correct?
No.
Brown hair is dominant.
Brown hair is dominant.
Then
It's like blue eyes.
I'm a mutant, yeah.
But there's mutations too, so we can't forget that. And that's why there's so much variation in the species as well. Remember, mutations don't have to be bad. Mutations just result in different. And different isn't always bad. And the only way that different is going to be passed into the generations is if you live long enough to do what? Reproduce and have offspring and pass it on to your offspring. So that's where all the different in humans, as we made more and more, we have variations in hair color and variations in eye color and skin color because of that. So, different types of crosses, different combinations of crosses. The first one, the easiest one, is something called a monohybrid cross. That's a cross between that purebred, what Mendel thought of as a recessive parent, looking at the characters of some of the other parents and their offspring. These stupid things. So look at this little, so big S is dominant and little s is what? Recessive. Correct.
Yeah,
High school,
square.
high school level.
So don't do that. Okay. So because Mendel was not only a scientist, he was a mathematician too. He was able to take this information and apply some math principles of statistics to it. Now he knew if he had a purple flower, he had 2 copies of information that made that flower purple. And he knew if he had a white flower, he had 2 copies of information that made that flower white. Now the information he wasn't quite sure about, and this is where the crosses come in. So he took purple flowers that had been purple and forever and ever, and white flowers that had been white forever and ever, and decided, okay, it's time for these guys to mate. And what happened in the first mating, that F1 generation, is he created flowers that were all what?
Purple.
That were all purple. What's that tell me?
The purple is dominant.
Yeah. Well, we'll see why purple's dominant in a minute. But then when he took those guys, the F1 generation, and crossed them, he found something else out. That the white didn't go away, it came back in the offspring. So he thought that, okay, if it's not going away, it doesn't disappear, there must be separate little entities that are passed on for it to reappear again in the F2 generation. So he saw that 3/4 of the plants from the F1 generation cross were purple, but he also found the white came back. So, why? He developed 4 different hypotheses from that cross. Listed here using modern terminology, including the word gene instead of heritable factor. We now call it the alternative version of genes called alleles. So alleles, we call the little characters or bits that were being passed on in his plants. In that monohybrid cross, each inherited character, an organism inherits 2 alleles from one parent or from each parent. An organism is homozygous for that gene if both alleles are the same, and an organism is heterozygous for that gene if those alleles are different. Now, alleles, bits of information, can either be dominant or recessive. We apply that term if all I need is just 1 copy of something for it to appear. That's called a dominant allele. In order for the recessive allele to appear, for me to see it, I have to have what? I have to have 2 copies of it. And that's what a recessive allele is. So with the white color being recessive, and next time I'll bring more markers in. But when I have a recessive trait, I have to have,
Never mind. You have
There's a blue one over there.
where?
underneath the screen.
Oh, no, it's green under there. I thought it was blue too. It was just a shadow. Okay. So I'll do this really quick. And we're looking at white versus purple. So P versus W. In order for me to be white, what do I have to possess? It's a recessive trait. 2. In order for me to be purple, and in this case, we knew this guy was a true purple. Oops, jeez. 2 big P's. When I cross these 2 individuals, they can only give what to their offspring? 1 of these alleles. So what's the F1 generation look like?
Big P, little W.
Correct. And what color are they? They're all purple. So this guy's purple, and this guy's white. Yes? And what happens when I cross the F1 generation?
Big B, big W.
And watch this. If I have two of these guys, I have this. Yes?
Yes.
I can only give one to my offspring. So what are the possibilities that this parent gives to the offspring? Oops. And this is the Punnett square you just saw. So I'm showing you statistically what the offspring are going to look like by splitting up the alleles and bringing them together on the page. And what's the possibility that this parent can give? Same thing, big P or little w. Now what do my offspring look like? I'm going to make them the same color for big P, W, big P. Do you see where the white one comes from? So these guys, because they possess at least one P, are all going to be what? Purple. There's my 3/4 purple. Yes? These are all purple. This guy's what? White. There's my what?
Your recessive gene. 1/4.
Yes? Gametes carry only one allele for each inherited character. That's your reproductive cells. So these guys represent the reproductive cells from these parents. Yes? Because they can only have one copy. They can have a big P or they can have a little w. Or they can have a big P or a big P. Yes? The statement is called the law of segregation. And why do we think we call it a law? Because it's consistent. It's been proven over and over and over and over again. So we'll pick up on monohybrid crosses in class on Monday. I will now go, and you're going to come with me, open your quiz. Quiz on Chapter 8. When is it due?
Monday.
Your quiz is due Monday at 1:55.